hand for something.
Oh, I should let
Sorry,
you know.
I didn't want to dominate the discussion. That's why I put it down.
Please, it's probably better if you just start yelling. I am on a Linux computer, and since this is a Microsoft program, they don't give me the hand raise function. I can't see if your hands are raised.
Okay.
Sorry about that.
So, could you help me understand the operculum, I think it was?
Mm-hmm.
And that versus operculate fungi and inoperculate fungi. What's going on there?
Let me find a picture. Come on. Did they give us a photo or like a drawing even? No, okay. Well, easiest way to think of it is imagine like a plant cell, like a little box, and you've got a fungal body growing on the inside. Okay, how does it release its spores?
So, from what I could gather, sometimes it has like some sort of channel that breaks through the cell wall, and it releases the spores through the channel. And then I wasn't sure how that was.
That channel is the operculum. I don't know if you
Okay.
can see this or if it's all blurry or whatever.
Yeah, I can see that.
Yeah.
So that's if it's an operculate fungi, they go through the channel. So how is that different from the inoperculate?
The inoperculate just busts out. Kind
Is it
of
like a
explodes
virus?
out. Yeah, kind of like a virus. Yeah.
Okay.
There's a million things you could memorize about all these different chytrids, and honestly, we don't even know all the chytrids that are out there yet. It's probably one of the most understudied areas of fungal biology is chytrid biology. They tend to be in weird spots. They're small. They're kind of hard to find. We don't typically know how to grow them in culture, so they're hard to study. Only a few species are we good at growing, and so the number of species that the book gave you was what, like, all told, maybe 700 species. Those are just the ones that are described. I've got a friend at UC Davis who studies these, and he tells me that there's probably 20,000 different chytrids, just off the top of his head guess, based on his molecular work. And trying to get them to mate together to see if they're the same species, but it's a lot of work. So going into chytrids, we're already kind of at a disadvantage. Like, we're studying something, yeah, we can write book chapters about it, but it's pretty limited. And this book is from, I don't know, 2002 or something. So it's a bit out of date. Okay. But did I get your operculate question?
Yeah, I got
Okay.
that.
I've got a question about one of the techniques they used.
Okay.
It was the part where it talks about maximum likelihood bootstrap analysis.
Oh, in the tree, the paper?
Yeah, sorry. My biggest question, yeah, I just had a hard time understanding which phyla they were taking the genes from. I understand they were comparing genes, but I just kind of got lost when they were talking about that part. I just didn't know which phylum they were taking them from, if that makes sense.
Which phylum? If you look at that paper, the tree there that they've got, that's kind of split up into two chunks. Every species on there, they took the same genes, the same 6 genes from each species.
So those 6 genes were from every single species.
Every single taxon that they looked at, they took those 6 genes and then they stick those genes all together, pretend it's one big long gene, and that way they can align them between all the different species and then start building their tree. Now, a maximum likelihood tree is just one way, a mathematical Bayesian tool for finding which tree is most likely given the data. Okay, and the idea of a bootstrap analysis, the little numbers on the tree, like at each node, they'll have two numbers there, like 100/93, right? 100/81. That's showing the percentage of times that node was resolved when they built the tree. Maybe I should back up a little bit. There are, for a tree this size, an astronomical number of possible ways to draw relationships between all these organisms. And I mean, we're talking like 2 × 10⁻⁵ right? So we build a short little hypothetical tree and use that as a starting point. And then we randomly rotate and swap nodes and then calculate how likely that tree is given the genetic data. And we give it a score. Okay, and if that score is better than the previous iteration, we keep that tree and then start randomly swapping around stuff, calculate the score. If it's worse, we throw that out. If it's better, we keep that one and do it again. In this case, typically people do this like 40,000 times. I don't know how many they did off the top of my head, but then those little numbers show the percentage of the final trees that had that same relationship. So it's kind of like a confidence value.
Okay, that makes more sense. Thank you.
Mm-hmm. It helps to remember that every phylogeny you see is a hypothesis.
Hey, Dr. Zahn.
Yeah.
So I was just late. There are 2 meetings going, and there's 2 other people. I'm not sure how to get back to the other one, but we couldn't figure out how to get into this one.
There's 2? Okay.
Yeah.
Let me.
Yeah,
I
I
don't
was one
see
of them.
it.
I made it here. So.
Okay.
Oh, it's in the general?
Actually, I don't know. I went to both, and so.
Yeah, the other one was in general.
I'll be right back. Let me pop over to that one.
Okay.
Has he been recording this?
Okay. There was nobody in there, so.
Are you recording this so that we could go back to see what we missed at the beginning? All right, thanks.
All right, so yeah, the meetings will always be in the live stream channel for this team. All right. Okay. Other questions about the methodology of the paper, their conclusions? Some of this is probably new. I don't know how many of you are familiar with building phylogenies, or if you've done it in the past or know how they work or how to interpret one. Yeah, can you
type a question in the chat? Are you able to see that?
Yes.
Okay.
Yeah, if you prefer not to speak and you want to throw a question up in the chat, that's fine. So a lot of times what they'll do with a phylogeny like this, particularly in this paper where they were interested in a morphological trait, right? That's what they set out to do, was look at where this trait turned up and calculate how many times it's been lost. They build their phylogeny and then they put a cute little symbol next to each taxon so you can kind of see which of these have flagella, right? And so that's where this becomes a useful tool, is seeing where the taxa that have flagella are and whether it's a monophyletic or polyphyletic group. And they see here that it's definitely quite polyphyletic. And so even some things that we used to consider zygomycetes or should be considered zygomycetes, are actually chytrids because they have a flagellum, like opidium there, and they call it Chytridiomycota even though it's nested firmly within Zygomycota. All right, what about the chapters?
So I've got just kind of a general question, because I was one of the ones that was stuck in the other chat. And so basically, I just want to understand what we're doing here. We're just asking any questions we have. Are we required to ask questions? Do we ask them if we, like, how exactly does it work? I came a little late.
You get 20 points for participation in this discussion. So that means answering or asking questions, contributing something of value or interest. Yeah.
Okay, cool. Thank you.
Yeah, Thursday classes are going to tend to be a bit shorter. I'm not going to have like set lessons where we go through slides and stuff. This is our chance to actually like talk to each other and learn about mycology apart from just, reading the text or following along with slides. This is, I think, where the interesting stuff can be talked about. Yeah. Or the stuff that you found interesting, at least.
Got it.
So did you want us to watch that video before class today, or is that for next Thursday?
Which video?
The video about the fungus and all the mushrooms and how the mushroom changed the world.
Oh, yeah, just before next week.
Okay.
Yeah. So not necessarily before today. The thing that you'll have to do, unless I tell you otherwise, is just do the reading and the quiz before Thursday.
Okay.
Dr. Sun, I have a question. I don't know exactly how to ask it, but I'll try to see if it makes sense. When scientists will usually look at a phylogeny and look at a morphological trait or whatever it may be that they're studying, do they always go back to the outgroup, or do they always like relate it back? I guess what I'm asking is, so they're looking at the loss of the flagellum, but do they always also go back to, okay, we're looking at this trait, what about relation to bacteria or cyanobacteria or the earliest ancestor of animals? I don't know. Do they look at that too, or not really?
It really helps if you do. I'm going to share my screen with you so we can look at this phylogeny. Okay. So right here where I've got my mouse, that's where the fungi begin.
We can't see your screen yet, just so you know.
Oh. Screen 2. How about now?
Yes, perfect.
Okay. So right here where I've got my mouse, that's the origin of fungi, the most recent common ancestor.
Right.
So all of these are fungi, but look at all these groups here. These are all their outgroups. So we've got metazoans, the animals, and they're showing that, yeah, they've got flagellated cells, okay. Choanoflagellates, mycetozoans, the slime molds here don't have flagella, at least that one doesn't, some slime molds do. And then plants. All right, so the importance of including this outgroup in a phylogeny is mainly so that we can what's called root the tree, provide it a root. If we didn't have these outgroups, we wouldn't have this particular node of fungi right there. We wouldn't see how all of these are related to anything else.
So do most of these fungi have an ancestral trait to them, or is that just certain ones?
Well, they all have some ancestral traits.
Right.
They've got chitin in their cell walls.
Right.
Heterotrophs, sexual reproduction. So they've got a lot of shared ancestral traits, but the point of this paper was they don't all have that flagellum.
Right.
And that was an ancestral trait.
Okay. That's because they resided in mainly water until they became were able to move to land, and then they had different mobility.
Yeah, I mean, we're talking almost 500 million years that they were aquatic and separated from the animals. But so this little branch right here between the common ancestor of animals and fungi is about 1 billion years ago. And then this branch right here represents another 500 million years of evolution. So this tree isn't scaled to show that. It's scaled to show the number of DNA substitutions.
Okay.
That's the length of the branch. Yeah.
Okay. That makes sense. Thanks.
Always look on a tree at the bottom. Any good tree is going to have some sort of scale bar for you, and whether it's substitutions or years.
Okay.
This one happens to be substitution. So it's kind of deceptive. I mean, we're looking at 1 billion years of evolution, or 500,000 years just right there, and then the rest is another 500,000. So it's kind of tricky that way. Yeah.
Okay. Thanks.
Mm-hmm.
So as far as this tree goes, the more they're finding, different fungus and stuff, are they constantly updating them? And then this one will kind of just become obsolete like other phylogenetic trees, but this is just kind of the most complete one that they have now.
It was the most complete one until 3 days ago.
And that was
Yeah, that was hilarious.
It like totally ruined our lesson, because now we've got a more complete, more recent tree. I stuck that up on our main general page. I'll go.
That one was.
I'll it up. This one hasn't been peer reviewed yet. But it's got a really cool, come on now, really cool tree in it. Okay, so you can see that it's not like all pretty. It's not been made for a journal just yet. It's kind of, this is what we call a preprint in science. So a lot of times now, if you've got really cool findings, you'll put it out in a preprint before it goes out to peer review, and this gives the scientific community a chance to kind of like make comments on it before you send it to peer review and improve it. So this may change a little bit, but we've got where's that cool tree? This is probably the best tree we have right now of fungi, at least it's recent. And the main difference is that in the paper you just read, they used 6 genes and they stuck them together. This uses entire genomes. Oh, so we have thousands of times more data that went into building these trees. What's cool about this is that we see a lot of the same relationships. And so those 6 genes did a pretty good job of predicting the full genome scale. If we zoom in here, there's our chytrids. Okay, so they're actually more or less kind of coming out monophyletic, similar to our paper here. We had Rosella like sticking out in its weird spot, and Albices. In this one, they don't go down to the species level. If they did, I bet those would still pop up here in the Zopagomycota. Yeah. So yes, like any tree, it's gonna go out of fashion soon enough. That one's been kind of the reigning champion for 14 years, and now this one's better.
So in this new tree that just came out 3 days ago, are there new classes to it, or are they? Or
no,
is it still just the same?
It's the same stuff. Yeah.
Okay.
And the issue here is that you have to have a lot of biological material to be able to make or to get the good quality genomic DNA. And so essentially we're almost limited to things that we can figure out how to grow in a lab. And so there's still a bunch of weird fungi that aren't included on this tree because we just don't have full genomes for them. Okay. There is a big project underway right now. It's actually funded by the Department of Energy, called the 1000 Fungal Genomes Project. and they're trying to kind of work on that and fix it. The Department of Energy is funding it because fungi make really cool secondary compounds, and they can break down things like switchgrass or old newspapers and stuff and turn them into biofuels. So that's kind of why they're looking at them.
Right.
I had a question about this. So it talks about how originally, maybe this is how I understood it anyway. Originally they thought that it was just one, like the flagella was just removed one time, but now it's 4 to 6 times. So were they the first ones to kind of have that idea and research it, or was that a thing and they just kind of found it? And then
yeah.
And then are they like, could there be more and they're searching for more, or is that just kind of like they found out that that's how many there is?
They found out that it was at least 4.
So with further research, they could find that there was even more events that could have happened. Right. Okay.
I don't know that anyone is actively working on that at this point. I think kind of the interesting open question was it once or more than once? And they said it's more than once. And then everyone kind of said, cool, and then moved on to other things.
That makes sense.
I haven't seen, maybe some lab somewhere and they just haven't published yet, but I have not seen it yet. Maybe I just haven't found it. I doubt that anyone would spend the tens of thousands of dollars and years of work to prove that it was actually 6 times, not just 4. But maybe.
Okay, so the breakthrough is just it was more than one, and that's all they were looking for. Okay.
More than one. And what this paper really did was change fundamentally the way we thought about chytrids. We used to think that they were monophyletic, right? And so we treated them that way, and there were confusing things like why are some holocarpic and some enterocarpic, and why do they have these weird mating systems? Well, it turns out it's because they're a polyphyletic group, and it's just the stuff that has not yet lost its flagellum. Now, there's an alternative hypothesis that could explain this also, and that's that fungi all lost their flagellum, and then each one of these groups regained it independently. Doesn't seem likely to me, especially given that they have, if you saw that section of chapter 6 in the textbook where it looks at the flagellal structure, like the ultrastructure, it's all the same in all of them. And so for that to evolve independently exactly the same would be really weird. Quite unlikely. All right, what about these other fungal groups? What do you notice? Well, in terms of the number of species, right off the bat, we've got like basically all the chytrids here. We've got some zygomycetes in here, and then this is our dikarya group that we're going to get to know pretty soon. The Basidiomycetes, and this entire right side is ascomycetes. So extant fungi have undergone a lot of evolution since they've gotten onto land, just really rapid evolutionary processes. Well, I don't know if really rapid, 500 million years isn't super fast, but the more recent groups have a ton more species than the groups that resemble the common ancestor. All right. Questions about fungi in general, maybe, that the book or this paper brought up for you?
So I have a question just about fungi in general. So it talked about that there's more than just chitin. I can't remember all the components that go into the cell wall of chytrids. Is that the case for all fungi, or is that just chytrids?
There's some weird ones. When we get into the zygomycetes, some of these fungi that are like obligate insect pathogens, they have chitosan and weird beta glucans making up part of their cell wall also. But all the mushrooms that you know of and the bread mold and all that stuff, that's just chitin.
Okay, thank you.
So it does seem to be a more primitive, quote unquote, trait.
But do all fungi at least have some chitin in their cell walls, or are there some that just they don't have it at all?
The real question I've got is, should we call them fungi? There's a group called Microsporidia that seems to treat out with the fungi really well, but it has almost no fungal characteristics. It's the smallest eukaryote. It's actually an intracellular parasite. So it's the size of a bacterium, but it's a eukaryote, and phylogenetically it looks like a fungus, but it doesn't do fungus stuff. Yeah. But for the purpose of this class, let's go with, yeah, all fungi have chitin.
Okay.
All right.
Will we be going over our quiz questions at all now that the quiz is over?
Yeah.
Just to make sure we do have the right answers?
All right. I'll share my screen again. All right, got it?
Yep.
All right, so we've covered question 1 pretty well. What evolutionary event were they interested in? And that was loss of flagellum. Okay. Question 2. They named a new monophyletic clade the euchytrids. What does that mean about traditional definition of chytrid based on morphology?
Would that be that in light of new evidence, you can't just base everything on morphology? You need stuff like molecular evidence, DNA type stuff, enzyme products, that sort of thing?
Yeah, and what it really means is that it was polyphyletic.
Polyphyletic, yeah.
Yeah. So you could have answered that question with just it was polyphyletic. Okay, yeah. And then what new information is shared about the evolution of fungal flagellum? Multiple losses. Okay. Was the common ancestor of all fungi adapted to marine or freshwater?
Marine.
I'm just confused about this.
Wasn't it unclear?
Yeah, the answer is we don't know.
Okay.
And the paper actually pointed that out pretty clearly. They said we can't tell based on this. All right, what 2 phyla are found in Dicaria?
Mycology and Basidiomycota.
Yep. Okay, taxonomic framework for classifying fungi prior to the 1980s.
Morphology, basically, looking at them.
Morphology, yeah, we're looking through a microscope. If they look the same, we put them close together in a tree, and it's transitioned towards phylogenetics since then. All right, did everyone, I think everyone got this. Okay. And then, how do you tell if a chytrid is holocarpic or eucarpic?
If it's holocarpic, it has a thallus that's just like a mixture of reproductive components lacking vegetative components. And if it's eucarpic, it has both reproductive components and vegetative components.
Yeah, so holocarpic, the entire fungal body basically turns into a reproductive organ. Yeah, and so it's a big blob, and it sits, inside of a cell, it absorbs stuff, grows in there, absorbs stuff, and then it turns into a zoosporangium that releases those flagellated zoospores. Eucarpic looks more like a ball with threads coming out of it that go down into the stuff. So that's the vegetative part. It goes and eats stuff and feeds the reproductive organ.
Dr. Zahn?
Yes.
Could you basically just say that the presence of rhizoids makes it eucarpic, or is that too?
Yeah, I think that would work.
Okay.
Yeah, because holocarpics don't have them. Okay. What's a zoospore?
Zoospore is basically an algae that's capable of swimming with the use of a flagellum.
Did you say an algae? Could
or,
be.
Well,
Isn't it just a fungi that can move in the water?
It's any spore that can move around.
Okay. I mean, I read something that said a spore of certain algae, fungi, and protozoans.
Yeah, okay.
So that kind of all within that category,
so
I guess.
Any spore that can move actively on its own is called a zoospore.
Okay.
Zoo meaning animal, because animals move, right? And so it's a moving spore. And so, you'll find them in ferns, some algae, protists, and in the chytrids. Yeah. And what do chytrids have in common with other fungi? Well, chitin. Sexual reproduction.
They're heterotrophic.
Heterotrophic. Yeah.
Do you want us, for questions like that, do you want us to be exhaustive, mention everything or just a few things?
Yeah, just a few things. That's fine. Okay. Questions about?
Yeah, I got
do you?
Is this how they're generally going to be set up, that they're mostly fill in the blank, maybe a couple matching or multiple choice?
Yeah, just a mixture, probably tending towards fill in the blank.
Okay.
Yeah.
Do you post the scores of our quizzes on Canvas so we can see where we're at?
I haven't graded them yet because they were just due.
No, yeah, I'm just asking, like, in the future.
Oh, yeah. So right now, I think everyone's got 2 points because everyone got the matching one, right? And the rest I have to grade. So, yeah. We'll post those.
I mean, Professor Zahn, it would just be so much easier on yourself if you made them all multiple choice.
And it's pretty boring.
I agree.
Yeah. I want to give you some creativity. Not all the questions are going to have a set answer either. Like the tricky question here, were they marine or freshwater? Right.
So for that one, the reason I said marine is I was reading in the textbook, and they said something about, like, there was fungi before 900 million years ago, before terrestrial anything had ever developed. So I was like, well, they couldn't get to the freshwater if terrestrial stuff didn't develop, so it had to be marine.
I see your thinking, but if you think a little more closely about this, those fungi weren't necessarily chytrids, were they?
No.
Right. And so the question was, according to the paper, was the common ancestor of all chytrids freshwater or marine? And they clearly said, we can't tell if it was freshwater or marine based on our evidence.
Wasn't it because all their findings they did in freshwater anyway?
Yeah.
Okay.
Of course, the common ancestor of all fungi was marine. So you're right about that. Because, I mean, that's going right back to, where animals and fungi split. 1 billion years ago, there was nothing on the land to speak of bacteria, right? So common ancestors of all fungi, definitely marine. Chytrids, we don't know. All the extant chytrids right now may have diversified after coming onto land. All right, any other questions about the methods that they use in this paper? They got pretty esoteric if you're not quite familiar with this kind of thing. No? All right. So, class news. little housekeeping here. We have got all your plates made, so we will have those available for you starting Monday, and you can go to SB 242. Sometimes I call it SB 243 because it's basically the same room. It's the main biology office, and you can pick up those plates starting anytime Monday morning.
Are they just at the front desk, or?
Yeah, they'll be in a box in the front desk that just says Mycology.
Okay.
Probably a note that says, if you're not in Mycology, don't steal my stuff. So in there are a few things. There's a plate that's kind of already sealed up with parafilm, and it's got some Ascomycetes growing in it. Just leave that alone. We'll get to that when we look at mating crosses and stuff, and everyone has their microscopes available. There's a little tube that just says pollen on it. If you open that up and wave it around, all your pollen will disappear, so be careful with that. That's how we're going to find chytrids. Okay, so they're actually not too difficult to find. The freshwater chytrids like to eat pollen, and so I've got some ponderosa pine pollen in there. It doesn't take much, just a few grains, and so I just kind of tap some in to each of those tubes. Your job will be to find some water outside, not like tap water or whatever. So go to a little stream, drainage ditch, lake, wherever you want to find chytrids, and just drop some water into that and wait 3 days. Then you'll be able to look at it with your scope.
So everything that we need to pick up, is it in like a little packet, or is it like?
Yeah.
Okay.
It'll be in a packet.
So, all right. So we don't have to just go say, all right, did we get one of everything already, or?
Right. I think I've got like a giant stash of old grocery plastic bags here at my house. I'm just going to throw that, tie it up. Yeah.
There you go.
So grab your grocery bag and you're good to go. I realize that not everyone has a microscope yet. It's on order, and the Postal Service is apparently being dismantled, so it's taking things a while to show up. So I'm not going to put a super hard deadline on the chytrid assignment, but as soon as you get your packet and you have your microscope, go ahead and add water to your little pollen tube and take a look at it. That assignment, if you see on Canvas, let me find it here. Oh, I don't even have it posted yet, but that's just going to be post a photo of a chytrid that you find. Okay, so pretty straightforward for 10 points. And what I expect you're going to see is a little Mickey Mouse shape, and then a blob on it.
Are these assignments just actually under the assignment tab, or are they?
Yeah, that one I don't have posted yet.
Right, but in the future they'll just be under assignments. Okay.
Mm-hmm. So this is what you're looking for. I'm a great artist. The Mickey Mouse is the pine pollen, and then you're going to have a chytrid sitting on the top of it with some rhizoids coming into it, eating it. So what kind of chytrid is this?
Eucarpic?
Eucarpic, yep. So that's what we're going to find. That's really common all over the United States in surface water, and so they just kind of hang out waiting for things to show up like pollen, and they eat it. Chandler, you had a couple photos of chytrid stuff, right?
Yeah, they're actually going to be pictures of tadpoles' mouths. So I did an internship in Brazil last summer where I was essentially researching the effects of BD on keratin-based structures in tadpoles. I can try to pull them up here.
Yeah, share your screen with us if you can.
Sure. probably start with a healthy one. Can you see my screen?
Mm-hmm.
Okay, cool. So this is going to be a tadpole's mouth. They're laying on their back. This is not the easiest thing to do because one, it's with a dissecting microscope, and two, tadpoles don't like to be out of water.
Right.
So sometimes they're not the best. This is probably a really, really good one where you can see the individual keratin structures here. Essentially their teeth forming, and you can see this is a healthy tadpole that hasn't been infected yet because their keratin structures haven't been absorbed, eaten, attacked. So that's essentially what we would identify as a healthy tadpole. And then these are going to be some examples of one that we saw that were infected with BD. So you can see that a lot of them have been disintegrated. They're out of shape. This one's also better. And also just looking at the tadpole itself, it's going to start sloughing its skin, sloughing all those keratinocytes that the BD is attacking, the zoospores is attacking. And it was like very obvious when a tadpole was going to die. Usually they died once they were infected. Typically we gave them about a couple days to live, and then they would die off. So it was
fast.
it was a fast turnover. Also, they were kept in aquatic tanks, and we would capture them from a riverbed nearby in the rainforest. So obviously the water wasn't best. It was in the middle of like a field research station, very poorly funded. But yeah, so this is kind of what I did. I took pictures of tadpoles' mouths for a really long time. We were looking at infection rate, and also they were doing a long-term research project on how weather and climate change affect the concentration of zoospores in the nearby bodies of water.
That's awesome. So what she showed us there is chytrids killing amphibians. And I mentioned this briefly during our lesson last class period, but this is a massive problem all around the world. So the BD is a chytrid disease named for this, I have a hard time pronouncing, Batrachotrium dendrobatitis. So it's a chytrid, and it does eat keratin. And so if you want to find these in the mountain lakes here in Utah, you can. Keep in mind that you got to be really careful though, because you can spread this stuff around and it can wipe out indigenous populations of like tadpoles, frogs, toads, all kinds of stuff. So you can take fingernail clippings, it's keratin, and clip them into the water and wait a week or two, and they'll actually start growing on there. And if you see that, it's kind of a bad sign. Yeah.
So is chytrid affecting amphibians at the same rate as as adult frogs as they are the tadpole species? Because like 3 days is super fast.
3 days seems super fast. For larger, like fully grown amphibians, I don't think it's quite that quick, but it does kill them. It's like a near 100% kill rate. And so there's been some work done. Most of the work has been done in the Sierra Nevada mountains, but some has been done here in Utah in the Wasatch. And entire lakes have been completely wiped out from all their amphibians, just in a matter of about a month.
So I know this has been an issue for a long time. Are we getting any closer to a solution?
No.
Cool.
Yeah, uplifting, right? I mean, there's a very real chance that 90% of all amphibians species on Earth will be dead in the next 50 years, extinct, because of this. Happy Thursday, everyone.
I had a question.
Yeah.
Learning about the chytrids, it said that they can be they can be biotrophs. They can be saprotrophs, and they can also be pathogens.
Mm-hmm.
Is that something that's common among all fungi?
Yeah, fungi are going to fall into like main nutritional categories like that. A lot of fungi are biotrophic. They need a living host cell to get their nutrients. Typically, it's going to be no, it gets really confusing because some species, an individual species, can do either sometimes. And so, yeah, you'll find it all across fungi, this kind of like flexibility of ways of making a living, right? We'll see it even more when we start talking about medical mycology. We'll have a week where we discuss that because, well, we have fungi living on our skin and in our mouths right now, and they're fine, right? They're biotrophic. They need us to live. They actually help prevent disease in us, but then they can turn pathogenic on us if we let them, because they don't have like a sharply defined moral code. They don't care about us at all. They don't know we exist, and so they're happy to kill us if we let them. And so immunocompromised people especially are at really high risk of fungal infection. Yeah.
Oh, wow. Okay, thank you.
Yeah. Okay.
Do we maybe be growing any fungi right now, or are we just waiting for? I mean, my microscope's in the mail, right? But do we just wait, or is there anything we should be starting right now?
Yeah. So your assignment was the moldy bread. So get that started. That's up on our website and on Canvas. It's the assignment: grow a zygomycete. Okay. So that assignment.
I noticed there wasn't a due date on that.
No, there's not due dates on any of these, you know. We're going to be playing it by ear because fungi aren't going to grow exactly when we want them to or how fast. So it's not exactly that kind of class. Your only like set dates are for quizzes and exams.
So as far as that mold growing on our bread, is it best if we put it in sunlight or keep it in kind of a dark area, or what's best for it to grow?
Throw it in a drawer and it's fine.
Okay.
And on the Canvas assignment, I've got a little video walking through it. It's not difficult.
Right.
You throw some mud on a piece of bread, stick it in a Ziploc. And the reason I'm having you start that now is because when we start talking about zygomycetes, I want you to have something to look at. And it takes at least a week. Yeah. Okay, what other housekeeping do I have?
I had what I'm not sure is a dumb question or not on the zygomycetes.
Okay.
The variety of bread I used uses honey exclusively as the sweetener in it, and if I'm remembering correctly, honey has some sort of inhibitive properties on fungus. Do you think that will be a problem for growing the zygomycetes on it?
It will not.
Okay. Cool.
Because you're adding water to it. Now, this is actually a pretty common misconception that honey is like antimicrobial, because you can have a jar of honey and leave it like open in your basement for 1,000 years and nothing will grow on it. That's true, but that's because the osmotic potential is so low. Okay, it just doesn't have enough water, and that's what keeps microbes from growing. So if you add water to honey, it suddenly becomes a microbe paradise. The zygomycetes, they're gonna love it. And I know people who swear by like putting honey on a cut. You know, you scrape your hand, like it's antimicrobial. I'm like, dude, once your blood mixes with that, it becomes like a bacteria feeding ground. You should not do that. That's a really bad thing to do. Don't put honey on your scrapes. So yeah, just use triple antibiotic ointment. It's humanity's like greatest triumph over nature, other than air conditioning. Use it. Yeah. Honey's going to be fine. What might be weird for some breads is if they have like a lot of BHT or weird preservatives in them. That may actually reduce fungal growth. Wonder Bread is one of those. It takes quite a long time to get mold on Wonder Bread because it's so chock full of like antifungal stuff. So maybe avoid that.
Does wheat bread work okay? Or
yeah,
okay.
yeah. Almost any bread's going to be fine unless it's like the kind of bread that you make in a lab. You know the bread I'm talking about.
Right.
Yeah. So avoid those. Everything else will be just fine. If you really want to go hardcore, make your own bread and then use that. It'll mold really fast.
Ooh, fun. That's the kind I'm using.
Perfect. Yeah.
That work for nothing.
Right. I think that's all I had as far as like housekeeping. So for next week, you're reading Fisher et al. 2018. So if you want to get a jump on that, since we're not meeting, or you won't have your quiz on that till next Thursday, but this is about zygomycete, and in particular, how hyphae grow. But they're using zygomycetes as kind of a model for this. And so this is the idea behind that inflating circus clown balloon. Okay. This is an interesting paper. It's not a bunch of like weird genetic stuff, no phylogeny. It's lots of cool like microscopy pictures.
And that's just on the class webpage, right?
Yeah. I'm
not
thinking
in Canvas.
Right. It might be an easier read for you than the last one. And then you've got your chapters from the textbook, and those go over zygomycete stuff. As you're reading, you're going to see all those terms coming right back at you. They're going to start becoming a bit more familiar. Don't get discouraged and don't focus on those terms too much. Try to read for the big picture of the chapter. Okay, the terms will just kind of, they'll come with familiarity. I wouldn't spend time like memorizing them or anything like that. Cool. So we move on to our next group, the zygos, next week. Any final questions?
Yeah, so I know that last time you said when we read, we're not really supposed to be, highlighting or like doing any like real in-depth notes, which is obviously a new thing for me in terms of taking a class. And so I'm wondering, how are we meant to, I guess, prepare for an exam or like know what exactly to focus on while we're reading, besides just getting an overall idea?
Let me show you. You see my page?
Yes, I can.
Okay. For each week, I've got a little thing here in resources called main study topics. Just a simple little text document. That right there is kind of your roadmap for what the exams are going to be. And keep in mind that you're going to have, it's open internet, open book, notes, everything for your exams. Okay, but this is kind of the things to focus on, the bigger picture ideas that I'm trying to I'm hoping will stick around. So
okay, so kind of use that as an outline if we still like to take notes.
Yeah. Yeah.
Okay, thank you, Dr. Zahn.
Mm-hmm.
Do you base your exams similar to your quizzes, where it's mostly just like open, like pre-response questions, or are they more multiple choice?
Just a mixture. It's gonna be more like the quiz, more free response. I'm gonna have you draw stuff here and there. Like if I give you a figure that's incomplete, complete this figure based on X, Y, and Z, that kind of thing.
Okay, thank you.
Definitely more concept based, yeah.
Will our test be given through Proctorio?
No. Hell no. That is creepy. I don't approve of that service. Somebody else taking control of your computer and spying on you, like in your bedroom, no.
I don't agree with it either.
Yeah, I think it's evil. So no, we don't do that. And I don't see a point. We're here to like learn some stuff about fungi look at some fungi, and get excited. So that's where we are. Cool. Any other questions? I'll let you go.
Awesome. Thanks, Dr. Zahn.
Yeah, we clear on what needs to happen for next week. Make sure your scripts are in the mail. Throw some mud on some bread. Okay. Cool.
Thank you.
Thank
See
you.
you Tuesday.
Thank you.